Good morning. My name is Mark Tessier-Levine, and it's an honor to be here today. I've met many of you since I joined the university as president last March, almost exactly a year ago, and it's great to see some familiar faces in the audience today. I do know, however, that some of you are new to the university, and for those who are, I'd like to say a few words about the institution and our work here. So this is the Rockefeller University at night. And Rockefeller was founded 110 years ago, first as a research institute, the Rockefeller Institute for Medical Research, and it was the first institution in the US that was devoted exclusively to biomedical research. The Rockefeller University Hospital was founded 10 years later. It's over there, to the right of Founders Hall as you walk up at the entrance. And it was the nation's first clinical research facility. It's the site of many famous discoveries, including the discovery back in the mid-40s that genes are made of DNA. A graduate program was added to the institute in the 1950s, so we became a university, and our mission expanded not just to understand life for the benefit of humanity, but also to train the next generation of scientific leaders. In terms of size, we are, relatively speaking, a small institution. That's deliberate. We have 73 heads of lab, about 200 research and clinical scientists, similar numbers of students, about twice as many postdoctoral researchers, and if you add in all of the scientific staff and support staff, there are about 1,800 people here in total. We focus on many diseases, that's illustrated here, although the way we come to focus on them is really by letting investigators follow their noses and sniff out opportunities where they think they can make the biggest difference. So we recruit the investigator and enable them, but we don't direct their research. They direct their own research. And in fact, our philosophy has been to identify the most brilliant and creative investigators and provide them with extraordinary scientific and financial resources. And again, our philosophy has been to recruit the person, not the field. So we never say, well, we need someone working on diabetes, let's go get a person working on diabetes. What we say is, who is the best person in the world working in any field, and we bring them here. And we end up with people working on diabetes and other things, of course. And our unique open structure, we don't have any academic departments, which again is unique for an institution like this, encourages collaboration, and it empowers the faculty to take on very high-risk, high-reward projects. In fact, Rockefeller is known for its transformative research, opening up whole new fields, such as cell biology and protein chemistry. You can see on the right some of the accolades that our scientists have received. The level of accomplishment is just astonishing. About half of our faculty are members of the National Academy of Science, which must be at least 3 times more than any other institution in the US. And if you go up to the top of the list, you'll see that we have over the years, we've had 24 Nobel Prizes in medicine or chemistry. Most recently, last October, some of you may have heard of the sad story of Ralph Steinman, one of our professors, receiving the Nobel Prize 2 days after he had passed away. The 24 Nobel Prizes in medicine or chemistry makes Rockefeller the leading institution in the world. We have more Nobel Prizes than any other institution. Harvard has had 21 in its history, Stanford 13 in medicine or chemistry. In fact, it's more than most countries. And in fact, if Rockefeller were a country, it would actually rank number 4 after the US, Britain, and Germany, and ahead of France. Now, speaking of prizes, I'd like to mention just 2 very important ones. First and foremost, I'd like to recognize Rockefeller scientist Elaine Fuchs, who's sitting in the front row here, who spoke about stem cells at the Women in Science breakfast last October. And some of you may have seen last week that she was named the recipient of the March of Dimes Prize in Developmental Biology, the most prestigious prize in the field of developmental biology. So I'd like to congratulate Elaine. Elaine is a leader in the study of skin biology and cancer stem cells, and her research has shed considerable light on some birth defects, and so it's related in a way to the talk that we'll hear about later today. And as members of Women in Science, you should all feel a special sense of pride in this accomplishment, because Women in Science actually provided the funds that enabled the university to recruit Elaine to campus as the Rebecca C. Lansfield Professor. I'd also like to share some very late-breaking news announced today that Professor Titia de Lang, the Leon Hess Professor, will receive the Dr. H.P. Heineken Prize for Biochemistry and Biophysics of the Netherlands Academy of Arts and Sciences. This is one of the most prestigious international prizes. The last person to win it, in fact, was Ralph Steinman the year before he received the Nobel Prize. And also of note is the fact that Titia is the first woman to win this prize. So congratulations to Titia. There'll be a press release today. Now, as you may know, this year we're celebrating the 15th anniversary of Women in Science, and to mark this milestone, contributions to the initiative this year will support the creation of a fund for interdisciplinary research on breast cancer, and we're very grateful to the Helena Rubinstein Foundation and University trustee Marlene Hess for helping to launch this fund with a $1 million challenge grant. The Women in Science community has responded most generously. I'm happy to report that, including the challenge grant, we've raised a total of $2.5 million so far this year. So, congratulations. And we hope to reach the 3 million mark in support of breast cancer research by the luncheon on May 10th. We're almost there. Now, on to today's program, a pattern for life. How a single cell sets the stage for development. I'd like to welcome and introduce our guest speaker, Dr. Kimberly Mowry, who is the Robin Chemers Neustein Professor of Biomedicine at Brown University. Rockefeller and Brown are both very fortunate to have the benefit of Robin's leadership, and who's been a wonderful advocate for science. I'd like to thank Robin as well. Dr. Mowry chairs the Department of Molecular Biology, Cell Biology, and Biochemistry at Brown, where her lab studies a fundamental question in biology: how certain key molecules are distributed and organized in egg cells. This basic question has far-reaching implications because the biological information that's stored in the egg plays such an outsized role in the development of the embryo. In addition to shedding light on the origins of birth defects, her research may also help us to understand some neurological diseases that are linked to the faulty distribution of important molecules within nerve cells. In the interest of time, please refer to your program for more information about our speaker. And now please join me in welcoming Dr. Mowry to the podium.
Well, thank you all for coming this morning. It's a real thrill to be here to talk to you about some of the recent research in this area. And it's also a real thrill to be here in this particular venue because I firmly believe that the mission of the Women in Science initiative is such a strong mission that's so important. We really need to support women in science, and as Dr. Fuchs has exemplified, women can do the most amazing science possible, and much of it has been done here at Rockefeller. So thank you very much for the invitation. So to start off with, when I talk about a pattern for life, and we talk about it starting with a single egg cell, what do we mean by a pattern for life? And what I mean by that is the embryonic patterning, that is how you put a head in a certain place and a tail in a certain place, and it really all starts with an egg cell. What I'm showing here are a collection of various egg cells, and compared to you or me or anyone in this room, these egg cells look surprisingly simple. And yet, for example, maybe I better use the pointer. For example, it may surprise you, but this is a human egg cell, but it doesn't look very different from, say, one of these other egg cells. So what is it about these egg cells then that can engender the patterning that's going to make a unique, highly complex, highly patterned individual? So again, if we look at this collection of egg cells and we see how these individuals here on the right are highly complex, it's very difficult, I think, to get your head around the idea of how we can go from this such a simple cell, shown as here, to this very, very adorable baby, shown on the right. Well, it starts with the egg, goes through embryonic patterning, I'll show you a little bit of that, and finally to the final organism. But again, these organisms here on the right look very different from one another. None of us would have any problem if I said which one's the baby and which one's the mouse, right? It's easy to pick out, but can you tell me which one is the human egg and which one's the mouse egg? Can we even tell which one is the egg of a tunicate or a sea squirt? Probably not, because they look all so different from one another. Now, in one case, we can probably pick out the egg and the organism. But that's not in particular because the chicken egg looks very much like a chicken. It's just because we know what a chicken egg looks like. Okay? There's nothing about this egg that really tells us, oh, that's where the beak is going to go. That's where the feet are going to go. And that's something that I'd like to explore a little bit today. So just so you know, we can match these up. We see here's a frog and its egg. Here's a worm and its egg. Here's the egg that didn't make this baby, but this is a human egg, and here's a baby. This is a sea squirt, and I think it's really amazing that this sea squirt egg looks almost exactly like a mouse egg, and yet the mouse and the sea squirt couldn't be more different from one another, could they? And yet somehow the information to make this mouse is here in this egg, the information to make the sea squirt is in that egg, and even though they look very much the same to us, what's inside of them is decidedly different. So how does that happen? Well, I want to look at this a little more closely using 3 different examples. So here is an egg of a worm, an egg of a frog, and an egg of a fish. And I also want to talk just a little bit about what I mean when I say embryonic patterning. So when we think about embryonic patterning, we really think in terms of what are called the embryonic axes. And so even though a frog egg looks very simple, we see here, we can easily tell where the head is and where the tail is. We have a back and a front, a dorsal ventral axis, and a left and right. And even in the embryo this is a tadpole of the frog, again, we can see this same patterning. We can see the front, the back, the front and the tail, the head and the tail, and the back and the front, and a left-right axis. And in each of these cases, and what I'm showing here again is the frog, we start with this single egg, which essentially lacks any discernible patterning. The egg, in the case of the frog or in the fish, is completely spherical. In the frog's case, we can see that there seems to be a top and a bottom to it, but we still can't tell from looking at it exactly how we're going to get that frog. And so now what I'm showing here are these movies where we're starting, in the case of the worm or the frog or the fish, we're starting from a very simple cell, and then the cells are undergoing divisions. So you can see the cells are dividing over and over again. And so we're going from this, starting from this single cell, we start with 2 cells, we go to 4 cells, and during that process, those cells are becoming different from one another. So when we go from the 1 cell to the 2 cell and the 4 cell, each of those 4 cells may have a different fate. It's going to go on and form different sorts of tissues and tissues in different positions within the final embryo, within the final organism. So again, we can see here that now with this fish embryo, we can definitely see where, or we could see until I looped it again, we can see where the head's going to be and where the tail is going to be, and yet when we started, we couldn't tell that. So how is it then that these simple organisms, that these simple cells, a single cell, can form such a highly patterned organism? So we can go back then to fertilization, and we start to think about what are the contributions? How do we get a fertilized egg? Okay, well, the way we start with that is we start with an egg, which again is a single cell, and a sperm, which is also a single cell. These two are going to fuse. The egg provides its maternal DNA, and the sperm provides the paternal DNA. Those fuse to make the zygote, and here we're looking at a zygote, and here one of these is a paternal has the paternal DNA and the maternal DNA. And if we go back to this image here, this is an egg being fertilized. You can see the sperm decorating the egg, and notice the size difference between the egg and the sperm. The sperm are really tiny, tiny, tiny compared to the egg. The egg is 10,000 times the size of the sperm. So what accounts for that? Well, they contain the same amount of DNA, right? But the sperm is essentially a modal package of DNA, whereas the egg contains both the DNA and a very large cytoplasm, Okay, so what do I mean by cytoplasm? What's in the cytoplasm? So this is, by way of an example, this is a typical animal cell. So an egg cell is an animal cell. It's not much different than any other cell in many respects. The cell has a nucleus. And the nucleus is where the DNA is housed. The DNA has the hereditary information that's going to be passed down from generation to generation. And then outside, and then this nucleus then, shown in a cutaway view here, is surrounded by what's called the cytoplasm. So what's in the cytoplasm? The cytoplasm has various organelles, and one of which I'm going to point out here. These are called mitochondria. The mitochondria are so an organelle are contained within the cytoplasm, and they perform specific functions. And the function of the mitochondria is that they provide energy to the cell. So they power the cell. You can call it the powerhouse of the cell. And unlike any other organelles within the cytoplasm, the mitochondria are unique in that they also contain their own DNA. So the only two entities within a cell that have DNA then would be the nucleus, which carries the chromosomes from the maternal and the paternal chromosomes, and also the mitochondria, which, as I said, has its own DNA. And I'll come back to that in a moment. In addition, the cytoplasm also contains tracts called the cytoskeleton that I'll talk a little bit more about in some detail in a couple of minutes, and it contains messenger RNAs and proteins. And it's those messenger RNAs and proteins that are actually going to drive the changes in cell fate that occur during cell division that I showed you just a moment ago. So just to explain a little bit more about the relationship between DNA, RNA, and protein. So you all probably know that DNA is the hereditary material, and DNA is carried down from generation to generation, can be copied and made another copy of itself. But DNA can also be decoded into what's called messenger RNA. And messenger RNA carries the information of DNA, and it's actually, this is copied in the nucleus. It's exported out into the cytoplasm. Okay, so if we go back, the DNA is copied here in the nucleus. This mRNA that we're making now is put out into the cytoplasm. And there in the cytoplasm, the RNA is then translated into proteins. Now, so the cytoplasm carries messenger RNAs and proteins. The messenger RNA can be stored for some period of time. And when you translate one messenger RNA, you can make many different proteins. Each protein will be the same. So each messenger RNA only translates into one specific type of protein, but each messenger RNA can be translated multiple times, and so you can have one messenger RNA and you can make 100 proteins from that RNA. Now, with the proteins in the cell, then perform the functions of the cell, but also when cells undergo differentiation, if one cell is making one set of proteins and another cell is making another set of proteins, that can then drive the changes that make the first cell different from the second cell. So now we go back then to this question of what's in the cytoplasm. So we have, again, the maternal DNA and protein, and we have the cytoplasm containing, as I'm showing in this cartoon, the red dots refer to maternal RNA. Once you have a zygote, the paternal and maternal DNA can then be expressed, and you can also make zygotic RNA, but what I'll stress is that these maternal RNAs are carried from the egg cytoplasm, and they're inherited by the zygote, specifically from the mother. And in addition, the mitochondria that are present in the maternal cytoplasm are also inherited by the newly formed embryo. And the sperm, in this case, then contributes virtually no cytoplasm to the newly formed embryo. All of the cytoplasm is maternal in nature. So these messenger RNAs and the maternal mRNAs and the mitochondria are all inherited from the mother. Now, as an aside, I'd mentioned that in addition to the DNA stored in the nucleus, that the mitochondria also have their own DNA. And so what that means is that in terms of inheritance, that the DNA is inherited from both parents, so both paternal and maternal DNA, whereas the mitochondrial lineage only is maternally inherited. And so you can see in this lineage diagram that the mitochondria are only coming from the mother. All right, and so what that has actually enabled scientists to do is to trace human migrations all the way back to Africa. So it can be traced actually by looking at the mitochondrial DNA. In addition, the inheritance of mitochondrial DNA also is the source of certain inherited disorders, such as mitochondrial myopathies. And these sorts of things tend to affect tissues such as muscle and brain that have high energy requirements. And so those also, so there are also inherited metabolic defects that can be inherited through the mitochondria. But I want to get back and spend most of the time really talking about this issue of the maternal RNAs that are inherited from the egg to the zygote. Now, in this case, what we've shown here in this diagram is we have one cell now dividing into two. And in this case, the contents of the cytoplasm are being inherited equally between the two daughter cells. But in fact, that doesn't always happen that way. And in many cases, maternal mRNAs can be asymmetrically distributed within the egg. So in this example here, a messenger RNA is specifically moved to the bottom of the egg. And that way, when this egg divides into two, we can see now that the daughter cells are going to be different from one another. So one daughter cell has inherited this information that's carried in the mRNA, and this daughter cell has not. And so when these are expressed, this cell then is going to express a different proteins than this cell is going to. And this can enable these two cells to have different fates. Now, an example of this that we can see is in this tunicate or sea squirt. These are organisms that essentially live on rocks in the ocean. And what you're looking at here is a cell of this tunicate or this sea squirt. And you can see just visually that the cytoplasm is different in the top and the bottom of this egg. And so this was dubbed the yellow crescent, and because this cytoplasm is quite yellow. And then as we go through cleavage of this embryo, we can see that this yellow cytoplasm, which is also called the myoplasm because it actually contains messenger RNAs in it that are going to determine muscle cell fates. And so this ends up being segregated to the future tail of this embryo. So we can even see now this embryo is now patterned. We can see here's the head of the embryo and here's the tail, and this yellow myoplasm has been inherited specifically by the cells that are going to populate the tail, and those in turn will specify that tail should have muscles. There are many examples of asymmetrically distributed messenger RNAs in egg cells and early embryos, and I'm just showing you a few here. So what you're looking at are micrographs of egg cells or very early embryos, and we've used, not we, these are images mostly from others. This is the only image I'm showing from my group. But what we collectively can do is we can use specific dyes to try and decorate these messenger RNAs so we can see where they are in the cell. So in this example here, we're looking at the egg of a fly. And this particular messenger RNA, this particular RNA here is localized to one end of the fly, and this end is going to form the head. And in this case, this particular RNA, if this RNA is absent, the fly will end up, well, you won't get a fly because it's not going to have a head. Okay, so these things can be really quite disastrous. And so other example, this is actually a snail. I'm not sure if you can see what you're looking at here. In this beautiful image is the RNAs are colored in red, and these are actually the spindles. So these are the dividing cells here, and you can see that it's decorating here at the end of a particular spindle. And what that means is that when this cell divides, that the cell on this side is going to inherit this RNA, and the cell on this side is not going to get any at all. And so these are similar images again where the RNAs are colored, and you can see that we have strongly asymmetric distributions of the RNAs in each of these examples, and there are many more. So I mentioned that this image is actually from my laboratory because this is a particular organism that we work on. And what we've done much of our work in the frog Xenopus laevis, and the reason that we work in the frog is that the eggs are very large and very easy to get. We can micro-inject them with essentially red labeled RNA and watch where it goes during the early cleavages. In addition, in particular, the frog is a good example of an organism where much of the early development is maternally driven. And so the frog doesn't even express the paternal DNA until many hours after fertilization. That's not true in every embryo, but it provides us a mechanism, it provides us a means to actually look very carefully at this mechanism of maternal inheritance. And so in this particular case, the RNA that you're looking at here, so what you're looking at here is a micrograph of an unfertilized egg. And the RNA that we've been studying is shown here in red, and you can see, I don't know if you can see the outline, but this is the outline of the cell. This is the top and this is the bottom, and the RNA is exquisitely localized to the bottom of the cell. And if we were to look much earlier in oogenesis, so oogenesis, I should define, is the process of making an oocyte, and the oocyte will eventually become the egg. We don't want to talk too much about that because it's not particularly relevant to today's talk, but an oocyte is the next step before an egg. So early in oogenesis, this particular RNA is uniformly distributed throughout the oocyte cytoplasm. So it's everywhere in the egg and in the oocyte. And then during the later stages of oogenesis, the RNAs actually move to the bottom of the cell, and then after fertilization, this is going to be inherited only by the cells that come from the bottom of the egg. And it turns out that if you misexpress, if the RNA is at the top of the egg, what happens is you end up not getting a tadpole, because this particular RNA is very important in terms of these patterning events that I spoke about earlier. So we were very interested, we and many others in the field have been very interested in understanding this process of exactly how do you go from here to here? How do you find a particular RNA and move it to a particular destination in the cell? And it turns out that the cytoplasm of the cell is highly complex and consists of what you might call cellular highways. And these are various images of the cytoskeleton. And you can see that there almost look like cables within the cell. And these cables can then be used as roads, essentially, to transport molecules and organelles within the cytoplasm. So let's take a closer look at these highways. So this is a great movie that I clipped from Biovisions, and what you're looking at here is formation of these highways, two different types. This is a microtubule. Okay, so I'm running it again. So this is an actin cable, okay? And what we'll see next is a microtubule. And so these are highways, again, that can be used for transport. But if you're going to move something along a highway, you need a truck, as it were, to move it. And so there are actually molecular motors that are present within cells. And these motors then actually can move on these highways and move cargo along the highways. And so there are three particular types of motors that have been highly studied, named dynein, kinesin, and myosin. And myosin, I'm not going to really talk about, but myosin has been shown to move RNAs on highways, and it moves RNAs on these actin highways. That was the first one that I showed you. And I'm going to focus a little bit more on kinesin and dynein. So kinesin and dynein are also molecular motors, shown below here in cartoon form. And these molecular motors can move on the microtubule highways. So if I go back now, if I can get it to play again, there we go. So now that I've told you about the motors, myosin moves on these actin highways, and kinesin and dynein will move cargoes on these microtubule highways. And so the other thing to know about kinesin and dynein is that they move in the opposite direction. So those you saw that the microtubules were forming from one end. Well, one end is different from the other, and dynein goes in one direction, in it's called arbitrarily the minus direction, and kinesin goes in the plus direction. So another cool movie from Biovisions, what you're looking at here, and when I talked about movies, these actually appear to walk on microtubules. So this is a kinesin, an image, a depiction of a kinesin motor dragging its cargo along a microtubule highway. And it's an incredibly cool image, but it really is there's been a lot of biophysical studies done on this particular motor, and this appears to be very close to exactly how this motor moves along microtubules. Now, one thing that's a little bit simplified in this image is that we're showing here one kinesin molecule pulling this cargo along. And in fact, it turns out that most cargoes are pulled by more than one motor. So in the field of trying to understand how RNAs are moving, we've discovered that, in fact, RNAs can be moved by both kinesin and by dynein. And in fact, probably there are multiple copies of each of these motors. Now, if in our system, if we block motor function, what we end up doing is we send the RNA to the wrong destination. So I hope you can see this, but I've also cartooned it so it might be a little easier to see. So when motor function is fine, the RNA is moved as it should be to the bottom of the egg, okay? Whereas when we block motor function here, the RNA gets moved in the wrong direction, and it's sitting right here. This is actually the nucleus of the egg, and the RNA is sitting here in completely the wrong direction. And that means that if we were to then, although this cell's fixed and not going to go anywhere at this point, but if this cell were to be fertilized and start to divide, the RNA would not be inherited by the proper cells, and development would be completely screwed up. So now we see that I mentioned that these two motors move in different directions. So you can imagine that this process might sort of end up in a war between these two motors, with dynein trying to pull it in this direction and kinesin trying to pull it in this direction. And it seems sort of like a futile process. But it turns out that, in fact, these motors are not actually acting against one another, but the whole process is highly regulated so that the motors cooperate with one another, so that one motor is active in one phase of the process, and the other motor is active in another phase. And by using two different motors with different directionalities, that enables the cell to fine-tune exactly where the RNA gets moved to. Because you could imagine, it's sort of like driving a car. If you can only go in one direction, you know, if you miss your exit, you're not going to go anywhere. And so this is sort of how we're envisioning this process, is by having the two motors, this allows the cell to fine-tune the process and put the RNA in exactly the right place. So I've been talking then a lot about how localization of mRNAs in eggs and oocytes is important for early embryonic patterning. But in addition, it turns out that mRNA transport in other types of cells is highly upregulated in motile cells, and it's also been shown to be upregulated in metastatic cancer cells. So this inherent, the ability for cells to move and for RNAs to move in cells, it appears to be very critical to polarization of almost every type of cell. And as Mark mentioned at the very beginning, a strong emerging theme in this whole area is the local translation of messenger RNAs in neurons. And so this process turns out to be important for synaptic plasticity and has been linked to a number of neurological diseases. And what's really exciting is that the mechanisms that we've been uncovering, these mechanisms relying on multiple motors and moving the RNAs to precisely the right point in the cell, turned out to be conserved in these other types of cells. So the wheel is not being reinvented in every different cell type. So the mechanisms that are being uncovered in these systems inform our understanding of these systems, and vice versa. So I want to end then with a gallery of mRNA localization patterns. I think that this is one aspect of this area that I find just incredibly beautiful, is just looking at these patterns. And so what we've seen here, I've talked a lot about localization of RNAs in egg cells, but as I mentioned then at the end, this is a motile cell, a moving cell, with an RNA localized here to the leading edge of the cell, and this is enabling the cell to move in this direction. And these are RNAs that are localized in neuronal growth cones, and this is even RNA localization in a single yeast cell. So I want to thank you for your attention and finish with acknowledging the people in my laboratory that have done much of the work, also research funding and the Robin Chemers Neustein Professorship, and thank you so much for your attention.
Well, thank you very much, Kim, for that fascinating talk. The floor is now open for questions, and there are roving microphones, and I'd ask you to please wait for a microphone so that we can all hear you. So questions.
Thanks so much. It was a great presentation. I have a very simple definitional question to begin, and I think I understood it just from the context, but what is the specific definition of upregulated?
Turned on, I guess, would be, perhaps turned on is a, upregulated is turned on, but what it implies is that we already have a certain level, a basal level of expression of whatever we're talking about. So what I was particularly talking about were RNA binding proteins that regulate the transport of these RNAs. And so there are a number of examples in metastatic cancer cells where the levels of these proteins are increased, specifically in the cancer cells. So it's not like they were off and then turned on, but they were low and now they're high. Sorry, too much jargon. It's hard for me to break my jargon habit.
Thanks so much for a very interesting talk. I was thinking, as the fertilized egg develops and the DNA material is positioned within the developing embryo, if something goes wrong and there's an interruption in the process, when does the cell know when to abort?
Why, that's actually different in different organisms. So for example, for many of the ones that I was talking about, many of them are free-living organisms. So in that case, there isn't really any abortion per se. If it's a frog, the embryo dies and it's in the pond and it just goes away. In terms of human development, not my area of expertise, but if these early patterning events are not done properly, in many cases there'll be a miscarriage. But I can't give you too many specifics about human embryology.
Thank you.
I'm a little confused about how the mitochondria and the cytoplasm fit together.
So the cytoplasm, you can almost think of the cytoplasm. So if we have a cell, the nucleus is in the center, contains the DNA, and it's surrounded by what you would like a watery bag of cytoplasm. So it's water, but it's got proteins, it's got RNAs, has these organelles in it. So I didn't talk about most of the organelles. The only one I mentioned was mitochondria, but mitochondria is an organelle. And these are contained within the cytoplasm. The organelles each have a specific function, and the function of the mitochondria is to provide energy for the cell. And in addition, it's complicated to explain, but the mitochondria are thought to have arisen from something called endosymbiosis. And so they are thought to have actually arisen from a bacterium being incorporated into a cell. And that's why in ancient times, evolutionarily ancient times, and that's why they're thought to have their own DNA. But what happens in the cytoplasm is that the mitochondria actually replicate, and they have their own copies of DNA, they have their own DNA, and it's separate from the nuclear DNA. So that DNA, it encodes proteins that are in the mitochondria, it encodes the translation apparatus for the mitochondria, and the translation can be accomplished actually in the mitochondria. And so because the mitochondria have their own DNA, they're unique from any other organelle within the cytoplasm. And they're going to be inherited from the egg cytoplasm from the mother. Did that make sense? Yes. Okay.
Thank you for that talk. I'm hanging on by a thread here.
Sorry.
When does the male, okay, changing this. On those highways and the movers, are they all also female? No.
No, so those, so yes, in this case that I was talking about, because so the motors are proteins actually. So they're made of protein. And they're in the cytoplasm.
So still all female.
They are all female during early development. So now later on, you have all the cells in, so your nerve cells, for example, they have those motors in your nerve cells. But the particular motors that are in your nerve cells are not ones that were inherited by the egg. Those are ones that are going to be expressed from your from your genome that has both the maternal and the paternal component. But the motors that are active in the egg, moving those things around, those are maternal, because at that point we haven't switched on the embryonic genome. Does that make sense?
When do the embryonic gene switch on?
So that really depends on what organism that you're talking about. Okay, so
What makes male DNA expressed?
So one thing about the male DNA is you saw how small the sperm was, and then in that picture, you saw that then when we had the two nuclei coming together, that nucleus then was much bigger than the sperm, even though that's the parental nucleus. So in the male sperm, the DNA is very, very tightly compacted. And because it's really balled up like a ball of yarn, it's going to take a little while before it can be expressed. Okay. But in organisms like humans, the paternal DNA is expressed very, very early, at least probably even before the first cell division. Okay, because human development occurs much more slowly than does something like frog or fly development. So in examples of something like a frog or a fly, those organisms, unlike a human embryo, which is nurtured within the mother, okay, those embryos are developing outside in the wild, wild world. Okay, and there are many, many predators that think eggs are tasty to eat, okay? So what needs to happen in those cases is that those organisms need to develop very quickly to the point where they can swim away, okay? So a frog or a fish. So what happens in those organisms is that the first several hours of development, we don't have the paternal genome being expressed at all. And it's all maternal. So in the frog, for example, till what's called the blastula stage. And that's about 16 hours. So in that case, the paternal DNA is turned on quite late. But again, other organisms like humans, the paternal DNA is expressed very, very, very early. Although even in humans, there are examples where RNAs are localized within the egg. And in that case, it has to do with the choice between inside and outside, but probably more than I probably should go into at this point.
Some neurological diseases that are diseases of this transport mechanism. Which diseases would those be?
So, for example, Fragile X mental retardation syndrome has been linked to defects in local translation of particular, they haven't identified particular messages, so the link is through a RNA binding protein that is responsible for local expression of messenger RNAs. Also, some neurodegeneration diseases like SMA has been potentially linked to processes of this sort. My feeling is it may in fact just be the tip of the iceberg, but the strongest link has been to Fragile X. And I should mention that this business about being able to translate the particular messages at a particular synapse is really important for cognitive function.
Maybe, Kim, I can ask a question. How much, in the example of the frog, it's very clear the egg has this polarity, and some of the other examples that you gave. How polar is the human egg, and how much does localized mRNA inheritance drive patterning in the human embryo?
So that's a really tough question to answer because, as you probably know, there's not much experimentation that's done on human eggs. But the closest relative that a lot of work has been done on would be the mouse. Okay, so in the mouse egg, it turns out that certain RNAs, in particular CDX2 RNA, is transported to the outside of the spherical egg. And so that then when those cells divide, those RNAs are inherited on the outside. And it's my understanding that that has a lot then to do with the decision of what's going to form the outside versus the inside. And the outside being the outside and inside decision there having to do with what's going to form the placenta versus what's going to form the embryo proper. But that's the major example that I know of. And I don't know that it's been looked at in humans, but my guess is it's probably pretty similar.
I wonder if I could just follow up on my earlier question. Sure. Even in the case of the frog organism, when does the developing zygote know that it's flawed and therefore it will perish?
When does it know it's
so
flawed?
if you have a flawed zygote, at what point does that destructive process begin, and how is it informed?
So in fact, with the frog, what happens is that the cells will go on and live until they run out of nutrients. So in the case of the frog has a lot of yolk in the egg. And so the yolk will be used and the cells will continue to respire and just be cells without doing, they will continue to divide as well. But the patterning is not such that you're getting any structures that will allow that to become a feeding tadpole. Mm-hmm. And so at some point, those cells run out of nutrients and they're simply going to die.
Thank you.
I'm wondering in frogs or fish if there's been any research that would trace this misappropriation of the mitochondrial RNA to disabilities of the limbs, to withered fins or feet or whatever, animals that live but have a developmental disability related to physical, arms or legs.
So I don't know of any research being done in that way so that you would actually have loss of limb in that case. In fact, much of the research on mitochondrial diseases has actually been done in humans, but there are also people using fly models to look at it. And so what happens in what happens in these cases is that because the mitochondria are defective, that they don't provide sufficient energy. So that's why in the cases of these diseases, the tissues in humans at least that are largely affected would be the brain. The brain requires a huge amount of energy, and also muscles. And so these end up being these, what are called mitochondrial myopathies. Many examples are in terms of muscle debilitation. But some of the ones that actually the prognosis is even more poor, results in death early on because it's actually problems with the brain. But to my knowledge, people haven't been really looking at mitochondrial inheritance in frog and fish, but there are some people, again, that are looking at this in flies, but in fact they're doing it more for trying to take a look at evolutionary biology to trying to trace patterns of inheritance. So again, in terms of the diseases, probably the most important work has really been done in humans, and that's unfortunate because it means what they do is that they have patients to look at, and so unfortunately, these are diseases that are out there.
Great. Well, it's now 9 o'clock, and so I'd like to thank Dr. Mowry again for sharing her insights with us.
Thank you very much.